Hi, Dr. Norashikin. My name is Tian Ning.
My name is Teng Jia Yi.
My name is Gheetaanjali Singam.
We are from Subgroup 3. This is our assessment for the oral presentation for the research poster with the title Effect of Different Bulking Agents on Fat Batch Composting and Microbial Composition Profile. So I will show our research poster, then I will pass to Geeta.
I will begin with the introduction. Due to improved living conditions, there is an increased demand for food, which creates a large amount of food waste. And large amounts of unmanaged food waste can be detrimental to the environment. Despite several measures taken by the Chinese government, incineration and landfills remain the main way of food waste disposal, which is unsustainable. A solution proposed is using fed batch composting, which treats food waste on site. However, there are some challenges to this method. Due to the high organic matter content and high moisture in food waste, it reduces the composting efficiency. Bulking agents are added before composting to modify the original substrate. Unfortunately, the effects of bulking agent types and sizes on the feed batch composting of food waste are not well understood. The study therefore focused on these three objectives: analyzing the impact of different bulking agent types on feed batch composting properties, investigating the impact of different bulking agent sizes on feed batch composting properties, and studying the impact of temperature in feed batch composting on the microbial community.
A very good day to Doctor and everyone. Today, in this session, I'm going to talk about the methodology in the article. So to begin with, the composting process was carried out using a fed batch composting reactor. As you can see in the figure 1, this is a schematic diagram of the fed batch composting reactor, which comprises of different parts and an operating control system. So first of all, the composting materials are first collected, such as green waste, wheat straw, sawdust, and bamboo charcoal. After that, the bulking agents are then added into the reactor. The food waste was fed into the reactor at regular intervals according to a certain ratio. There are 3 different types of experiments set up in the article. These experiments include to find out the effect of bulking agents type on composting. Next is to find out the impact of bulking agent sizes on composting. Then lastly is to find out the impact of temperature on the microbial community in the fresh batch composting. There are measurements taken during the experiment. These parameters include moisture, wet loss rate, OM, CN ratio, pH, electrical conductivity, seed germination index, GI, temperature, and moisture content. The experiment is undergone for a length of 30 days, where the measurements were taken in intervals of different days. After the experiment is over, the article then proceed into analysis. These analyses include the physicochemical properties, the microbial community analysis, and lastly, the statistical analysis of the composting materials. So from the experiment, it helps the article understand the effect of different type of bulking agents on composting efficiency. So from the bulking agent types, GW showed the most promising results, where it shows the highest weight loss, the lowest CN ratio, the good compost maturity, where it has GI bigger than 90%. Next, on the effect of bulking agent sizes, GW also shows the best results, where GW size at 3 to 6 mm. It has optimal moisture retention, best balance in OM and EC, and as well as the highest GI among the other bulking agents. And lastly, on the experiment of effect of temperature on microbes, we found out to be at 50 degrees Celsius, Firmicutes, proteobacteria, and Plasmodium has increased. Therefore, the higher the temperature, the better degradation of the lignocellulose. That's all my part. I'll pass to the next person. Thank you.
Alright, thank you, Caleb. So let's move on to the discussion of our study. We tested how different bulking agents, particle sizes, and temperature affect food waste composting in a fed batch reactor. First, green waste stood out as the best bulking agent. It produced the highest compost quality with a moisture content of 3.16%, beside wet loss of 85.26% and a germination index of 90.73%, indicating mature compost. Its porous structure improved airflow and moisture balance, helping microbes decompose waste more efficiently. Second, the optimal particle size was 3 to 6 millimeters. The size allowed enough air and moisture retention to support microbial activity. Smaller particles held too much water, while larger ones reduced microbial contact and slow composting. Third, temperature had a major influence on microbial activity. At 50 degrees Celsius, heat-loving microbes like Firmicutes, proteobacteria, and the fungus Rhamsonia become dominant. These microbes are crucial for breaking down complex material like cellulose, improving compost speed and quality. We also observed a microbial shift from Actinobacteria at 40 degrees Celsius to firmicutes and basidial mycota at higher degrees Celsius. In conclusion, combining green waste at 3 to 6 millimeters with elevated temperatures in 45 to 50 degrees Celsius produce the best compost outcomes. It improves microbial diversity, compost maturity, and overall efficiency. Overall, this method supports a low-cost, scalable solution for food waste management in rural areas. It reduces environmental impact, avoids the needs for transport and landfilling, and enables composting directly where the waste is generated. Alright, that's all from us. Thank you, Dr. Narashikin.